Hi, I'm Joachim Schmid. I'm the Vice President for Software Development at NanoString. And I'm excited to be here and talking about spatial informatics, spatial AI, and public data sets. It's really an honor to be here, and I'm joined NanoString 3 months ago, and I've spent 25 years in digital pathology, digital diagnostics, and I'm really excited to see how we can combine some of that knowledge with a lot of what the spatial informatics brings and the opportunities to really learn more about disease and what's happening in cells. So with that, I would like to start my presentation by looking at challenges and opportunities in spatial informatics. So I really focus on the software piece. I think it fits nicely with what Joe was presenting earlier about all the application side of things and the exciting new learnings there. What is some of the challenges and opportunities to really make that easy for the user, easy so the scientists can focus on the science and not on how to manage the data? So there's a tremendous amount of data. I think Joe talked about 1 to 10 terabytes per slide. How do we manage that? How do we make that accessible so you can really look at that? And usability from the software side of things, it's very key. I believe in software should be usable without reading a manual. We need to make sure that we kind of allow the user to really interface with the data and work with it. As we have, as I said, there's a lot of data there, so AI is the opportunity here to really look and find new insights and really get the full potential what's in the data to come it out and really kind of do that. That will create, and we're already coming out with a whole bunch of data analytics tools, but that's the opportunity. As we kind of create the data, there will be a starting point of how many analytic methods there are, and there are already quite a number with Genomics, but there will be many more. But at the same time, how do we enable that? How do we make it scalable so that storage and compute is not the limitation? And that's where the cloud comes in. How do we kind of make it easy to access the data and really build on it as you kind of adding more and more experiments to the study? And as my experience has been very much in the digital pathology space, I don't want to lose the connection to H&E and all the things happening in digital pathology, but more like how do we combine things? How do we go beyond H&E and really kind of pull these things together and create new insights as we combine what exists in pathology today with new methods like the Genomics and the SMI methods. So we'll go a little bit deeper in all these different things. One thing we are doing right now is building an ecosystem to address these challenges and to enable, collaboration across the, our system and other systems. On the left side, you can see there are instruments which are all providing data. There's Genomics and there's CosMx. There's also the encounter data. But then there will be also, or there are data from Illumina, which we are using today, but there could be other instruments providing data into the spatial biology portal, which you see in the middle, which is really at the center of how do we work with the data, how do we analyze the data. So there's a data lakehouse, a place where all the data gets stored and then made accessible, you can manage and you can operate within. I will go more into detail on that later. And the right side, you see all the different opportunities on data analysis. There will be some very specific Genomics analysis pieces and CosMx analysis. And then beyond that, and it's colored a little bit different because I want to point out, could come from partners. Not everything needs to come from us. I think an ecosystem enables innovation across. Our eye selection methods based on AI. There's lots of cool methods out there which are really identifying different parts on the tissue. Open source analytics. The open source community is coming up with new methods every day. I think it's difficult to keep track on all the innovations happening there, but how do we enable that to come together with the data and combine that? Then I think there's lots of digital pathology analysis we can combine. And then I think the last one is probably the most important one here is spatial AI. Have the data ready for AI. And then getting different methods, analyzing the data and come up with new insights in there. I also want to point out, to make all these things happen, one of the key things is to have the data in a standardized format so that it's easy to access and easy to transfer in some way. And here I point to Seurat as one of the formats and OME-TIFF, but there are many other formats which could come to be important in the future. Here's a slide which Joe had in his presentation to really look at how do we combine the spatial platforms and provide all the data to go into the cloud, make them available on the spatial biology portal. And then there's 5 main areas of, kind of like focus. One is just pure image analysis. They are all spatial, so they're images which need to be analyzed, and how do we interact with the images. Everything is happening in collaboration nowadays. It's, as Joe said, not send an email with an attachment and that's how you get the slide. It's not possible. So collaboration tools are key. You want to make sure the data is protected, but at the same time, it's easy to share data and work together on that. So collaboration tools will be one of the key focus areas for the spatial biology portal. And then, because we're talking about a lot of data, it's really important that we can scale the compute and storage. And that the cloud gives us that. You can scale, and on the compute, you can have the compute when you need it. You don't need to buy the compute for the one time you need it. You get the compute at the time when you need it, and then it shuts down again when you don't need that. So really scalable. Data visualization tools are key. How to visualize all the data in a meaningful way, and there's many innovative methods getting more out of the data, and I'm sure we'll see more on that. And then, AI and ML is everywhere in today's world. So it's also very critical for spatial biology, and I would like to say, we'll make the data ready for AI. Here's a picture on our informatics portfolio today. It's around the Genomics platform. It allows the different users to interact with the software in different ways. The pathologist will use the software to select ROIs and approve ROIs. The lab technician uses the software to really run the machine, set up the experiment, and so on. The biostatistician analyzes the data, looks at the different statistical methods, and comes up with the best way to analyze the data. And then the principal scientist can review the results and combine all the results ready for publication. To give a little bit more detail on that, here's a screenshot of the Genomics data analysis suite, which gives you from left to right a whole analysis workflow. It goes from looking at the slide to what's the history and the probes used in the experiment to the analysis tool, and gives you a whole bunch of different tools to combine. And the interesting thing, or what I feel is very key, is that we never lose track of where is it coming from. So how is the data on the right side connected to the left side, the spatial context, because I think that's all about, spatial biology. Here's the data analysis and data visualization tools. There are different complexity levels here, but I think it goes from some basic methods to some really, really sophisticated methods like the pathway analysis with bar charts, heat maps, and pathway overlay, which are really, really exciting to see that from your data you create. And so it's kind of your toolbox to work with the data. As Anna said in her presentation, one of the key fundamentals for NanoString is always to keep that multi-omic approach and to analyze samples with the right method, or if there are more than one, use more methods to that. And I think it's very exciting that we can analyze tissue with an RNA and protein and combine the data and bring that together and really look at it as one combined thing. And as Joe showed in his slides, it's here's depicted as multiple sections, but it also could be done on one section. So I think that's very exciting because biology is happening not in RNA only or protein, it's the combination of these things, which really gives us new insights. One of the interesting things over the last year was to work together with Illumina and really create an end-to-end solution between our system and their systems to really bring the data for genomics together and put our NGS pipeline in base space at Illumina and to combine it and make it available on genomics. And I'm also excited to say that at end of March we'll launch the full integration of the Illumina NextSeq 2K and will be on instrument to really create the end-to-end experience. So I think it's really one of the examples which shows how collaboration between different groups really makes life much, much easier. And we'll talk more about that in the panel discussion, where we have one of the colleagues from Illumina joining the panel discussion there. Here's an example about public data set and the Spatial Organ Atlas. Joe already talked about that in his presentation. It's very exciting to see all the different data sets being in the public domain and what's been done with that. I think that's the graph on the lower left. The number of publications around that is just amazing to me. Being new to the field, I'm just kind of like amazed to see how many publications we see around that and the new insights created by these public data sets. And we'll add more public data sets as we're going into SMI and so on and make them available there. I will talk more about that later. I think another interesting thing is really how we advance innovation by supporting the open source community, by supporting the community, but also developing some of the tools and make them available through the Bioconductor. If you're interested, I would highly recommend to go to the Bioconductor website and look at some of these tools, how they can help to analyze some of the data. The reason for me to really support that is really to enable the innovation that happens out in the open source community and really kind of see what's possible with the data we create and bring that together in one place. Here's a slide which talks about our CosMx publication, which was kind of describing in November how the chemistry, the system, the technology behind that, and really showing first applications behind this. But I think what's really cool is kind of like that at the same time we launched a public data set of 800,000 single cells and made them available for download. And it's very cool that we had more than 200 downloads of the data set, which is not a little thing because it's 500 gigabytes of data. So it takes quite a while to download and some commitment to download all the data. And I don't have examples today, but what I have seen from the community is a few examples where different groups have started to use the data to create some new visualizations. I've seen some cool examples where people used AI to visualize different stuff. And then I also know there's a few publications in preparation where people using the data set to publish and come up with new things there. So exactly what we want is kind of like support innovation, get everybody to look at the data and think about what could I do with the data. And we'll kind of publish more data sets as we go along this year until launch to really share what the data will look like as we're moving into the commercial phase of the product. But please look forward to more data sets being published. Now I would like to give you a sneak peek at our spatial biology portal. As I said at the beginning, usability is key. So really creating an intuitive workspace to manage studies, images, and results, which are coming from Genomics and CosMx, are some of the key requirements for the software. So it's really, very easy to manipulate amazing amounts of data. And, the scalable storage in the cloud will allow you to do that. And I said, it's not easy to just attach the images to an email, but the method within the portal to share data should be as easy as an email. I would like to share these 3 images or 3 data sets with my colleague somewhere else. It should be like sending an email. And it will create great collaborations within that. And then it gives you access to a streamlined analysis pipeline, which I will talk about in a second. So the user interface will give you lots of opportunities to manage the studies and combine data from different sources in multiple studies. And then when you're done all the analysis, you can consolidate all the data in one view and work on that together, but also then organize the data for publication so you're ready to publish on that. And one thing which I'm sure everybody is very interested in is kind of like how to analyze the data. And there's a pipeline orchestrator built in there. On the left, you see the the data available. Then there's a pipeline orchestrator you have in the middle, and the results on the right. And you can create your own pipeline. You can save them and just kind of keep using them, optimizing them. But then you can also add custom modules to really add functionality beyond what's coming with the system. It will have a first data set, a first tool set of analysis methods, but then I'm expecting many more things happening on the custom side and other methods which will be added to the pipeline orchestrator. At launch, we'll have a portfolio of methods to analyze the CosMx datasets, and it will go from quality control to normalization to UMAP and so on. So we'll be a really rich data set, but I believe having them all play within the pipeline orchestrator will be the key to really kind of get the insights on the data. Here's an example we're using today, machine learning, to really combine the outputs from the cytoplasm segmentation and the nuclear modes into one final cell label, which we then use as we move from secondary processing into tertiary processing. Just one of the examples to really showcase there will be so many ways to utilize the data and work on one piece, like here on the segmentation piece. Lastly, I would like to get into what do I see as the future trends in spatial biology informatics. And that's just one small set. I think there's many opportunities, but one really important one I think is spatial AI. As we're creating the data sets and AI is developing new methods as we speak and do new ways of analyzing data, we'll see the combination of these two things creating really amazing new applications, new ways of looking at the data, and hopefully new ways of understanding biology. What's in the middle is the federated learning topic. And federated learning is kind of going with the topic of spatial AI. As any AI method needs access to data, and we want to make sure that AI is protected, federated learning is a technology to enable AI without moving all the data to one place, aggregate the data in one place. You can keep the data in different places. And yes, it's the cloud, but it's different instances of everybody's data. And only transfer the insights, only transfer the learnings, and then make one machine learning model, which is really based on data from multiple sites without bringing the data together. I think as we're talking about lots of data, samples from different sites, federated learning could be really one of the enablers for spatial biology. Lastly, I want to, what Joe said in his presentation, how do we combine the data and make meaning out of that? So I see there's lots of opportunities to really create new ways of looking at the data as we go into multi-omics, and that's combining data from genomics and SMI, or combining data from RNA and protein, or all of these. I think we don't know, but I think as the data becomes available, we'll see a lot of methods that go beyond just a single few, but doing data analysis across. So that's just a few examples of future trends in spatial biology. With that, I would like to say thank you, invite you to join the panel discussion where we talk about some of these topics, and thanks.
Hello, welcome to the Spatial Genomics Summit Informatics panel. I'm Jason Reeves, Associate Director of Computational Biology at NanoString, and we've got a fantastic panel here to discuss the opportunities, challenges, and excitement around spatial omics and the ways in which it will grow over the next coming years. Today, I'm joined by Chris Mason, PhD, Professor at Weil Cornell School of Medicine, Dr. Ruben Dries, PhD, Associate Professor at Boston University School of Medicine, Dr. Kevan, Samiee, PhD, Senior Director of Open Innovations at Illumina, Dr. Margaret Hoang, PhD, Associate Director of Research and Genomics at NanoString Technologies, and last but not least, Dr. Joachim Schmid, PhD, VP of Software at NanoString Technologies. So Chris, I'm going to start with you, if that's okay.
Yeah.
So your projects and your team investigates a wide variety of different topics. You do work in the microbiome, you do work with astrobiology, but all of this is now sort of, you have new tools to play with. I want to know what you're really excited about in terms of projects that you can now tackle with spatial omics really taking center stage.
With spatial omics, now we can actually get a much higher resolution of some of the real strange changes that occur to the body in every place of medicine and even spaceflight. So we know that, for example, COVID-19, we've published several papers in the past year showing that tissues really are disrupted and cells even somewhat forget who they are and where they're supposed to be inside the organs, inside the body. So this has all been really enabled by spatial mapping technologies, where we can see with the highest ever resolution what's happening inside some of the patients when they die, unfortunately, at autopsy. But we're also applying it to really unique environments. So what happens to skin when you go to space, or spaceflight associated neuroocular syndrome is a damage to the eyes that astronauts experience. And so we're looking at the mice that have been flown that simulate this and seeing what happens to their retina. So we can get extraordinarily high resolution from human and mouse models from COVID all the way to space. It's been really extraordinary.
Thank you. And as these spatial platforms mature, what are some of the pieces that are in play there that are going to be absolutely enabling for those studies that you want to do?
I think the key elements are really, of course, the technology has to be robust and the probes work and the antibodies are working. But that is something that, NanoString it deals a lot with itself. So by the time we get it and start to run the machine, it works just fine. But really the computational biology, the ability to take terabytes of data per experiment and mine that data, understand it, contextualize it, compare it to other resources and reference databases is itself a challenge, but actually really exciting because it gives us a lot of power and a lot of data. So machine learning thrives when you have a lot of data, and we finally have high resolution, deep phenotyping and genetic data on these tissues and on these samples. So I think it's actually just the beginning of a really exciting, sort of ramp up of data and utility and understanding biology.
Yeah, and as the data grows, we're going to face new challenges. I want to ask Kevan a little bit about this, actually, because Kevan has led a group over at Illumina. They're obviously the leading platform for bulk and single-cell sequencing, and I wanted to get your sense as to what opportunities Illumina is investing in to really enable spatial biology, and where you see Illumina sort of leading in that space.
Yeah, I think, the key area I'd say there's two key areas for Illumina and really enabling spatial biology. And the first one is just our continued investment in kind of our core sequencing platforms. We have platforms that really span, the output continuum, and being able to impedance match the type of output that researchers want with the type of experiments they're doing is really critical for us. So that's an area where we continue to invest. And I think, over the past decades, driving down the cost of sequencing bases has really enabled some of these applications to come to fruition. And then I think the second area is, as a company, we recognize that our sequencers generate just vast amounts of data. And so we've put in a lot of effort over the past 5-10 years to really build out platforms that enable people to manage that data more easily, starting with things like our Dragon technology, which, whether it's onboard the instrument or it's up in the cloud, it really lets customers, instead of having to worry about managing a bunch of FASTQ files, like going straight to their index count files or going straight to their variant calls, something that's a little less data intensive and a little more valuable at the end of their sequencing. And then we've also invested in our new Illumina Connected Analytics platform, which is enabling not only the really cool work that we're doing with NanoString to bring really seamless end-to-end solutions to market for customers, but also, enabling customers to go in and do collaborative data interpretation and analysis, really bringing together different omic data types along with clinical outcomes so that we can do more discovery and learn more quicker.
It sounds like you guys are keeping busy then.
If my schedule is any indication, then I would agree with that.
Well, all of that is going to be really critical as these platforms grow. The other side of the house, in terms of how these platforms take off, is not just the actual generation of data, but the management and interaction with it. So we've got Ruben with us. Ruben, you've been building quite a lot of tools in the spatial omics space. Can I just get your perspective on what opportunities you see there, what challenges the field might face, and what you're really excited about in terms of the new types of analytics that are coming online.
Yeah, of course. I think it's really challenging in the sense that with the spatial transcriptomics space, we have to kind of like merge different fields, right? It's not just only single cell. It's not just only image analysis. It's really, we need to think about how to create a hybrid scientist that can take all of that information and kind of like integrate it and make it meaningful so that we can also use it, for example, in the clinic or outside of the clinic. And I think that is currently the key challenge from a data analytical point of view.
And when you've been approaching this, what's your thoughts on how data scientists and computational biologists and bioinformaticians need to evolve to face some of these challenges that you're seeing right now?
Oh, well, there are many challenges. I think one of them is if you actually see the number of new methods that are being developed, we're really in this kind of like exponential phase. I think we're already or close to surpassing new methods, for example, for single-cell RNA-seq. What is difficult, especially for a user point of view, is how to use them. And so what some people have done, including us, is developing these toolboxes like Giotto and also Seurat and ScanPy, so that people can easily use that and do that from the beginning to the end. However, there is also this problem of interoperability, right? Ideally, you want to be able to go from one to the other and do whatever you can on all the different platforms. And that is, I think, a key challenge. The bioconductor community, they have this spatial experiment class, which ideally allows you to just use that class to store the spatial information, and then you can build any sort of method on top of that. So that is great. On a larger scale, there is a new group, it's called FOM, like Features Observation Matrix, led by Josh Campbell at BU and also Niels Gehlenborg at Harvard. And what they're actually trying to do is something even bigger that like overarches institutions, consortia, and really think about how should we represent the data so that everyone can actually access the data and knows where is the metadata, where is the cell locations, where is this, where is that. And if we can actually manage that and we can continue to use that, that would be absolutely great.
Yeah, it sounds like a great piece of work, and it sounds like the community is actively investing right now in thinking about these problems rather than letting them develop over time and create blockers for the ability to leverage these platforms. It's interesting that you mentioned some of the open platforms that have been developed in the past and the fact that we're moving even potentially faster than previously. On the other side of the house, Joachim has developed quite a lot of relationships in the digital pathology space and has, I think, quite a passion for making sure that images and metadata and count data is open and accessible. Joachim, what's got you excited about the future for spatial omics right now and the potential integration points of new data types that we haven't necessarily seen before in the past?
I think what got me really excited, I've been spending my career in tissue analytics, cytology, last 20 years, really to see how can we add to the tissue other information, meta information, a lot of information, and really get meaningful insights out of that. And I think I've spent, I would say, 15 years in standards for digital pathology, and it, kind of slowly moving. I think the earlier you can start thinking about standards and really defining data, the easier it is. The later you come, the more difficult it gets. So I think that's really exciting to get from the starting point into that kind of mode of can we standardize pieces and then also bigger ecosystems in that. So I think that's really, really exciting to me. And as Chris said, the mining of the data. What's the meaning of the data, and can we mine that tremendous amount of data and find some new things, new discoveries in that?
Yeah, and this is going to be a space that's going to grow, but it sounds like the community is actively engaged for once to really, really tackle this head on. In terms of the applications and the ways in which we are developing at NanoString, Margaret, I wanted to get your perspective since you're developing a lot of the assays that we work with. How are we thinking about engaging with the community at large, and what are some of the infrastructure pieces that we're tackling in-house that's going to enable this type of science to move forward?
Yeah, it's a great question, Jason. I think that with big data, informatics has always been the bottleneck. in all of the omics fields, and spatial biology is no exception to this. There are three main infrastructure needs, I think, in this nascent spatial field. I think of number one being this theme of the standardized data format. Pairing the image with the counts is really important. I know in the omics field, where many of the scientists might not be trained in the imaging, but I think pairing that image with the counts is really important because it creates opportunities for exploratory data that you might not be able to foresee when you're analyzing the data. So that's a really important part of it. I also think about the how standardization really revolutionized the genomics fields of DNA sequencing. I think of when the 1,000 Genome Projects with Richard Durbin leading that, co-leading that, and his postdoc, Hoang needing to align all the sequencing aligners and coming up with the BAM and SAM format really revolutionized that field. So I feel like the spatial biology field is sort of in the same nascent way looking at those community formats. Number two is sort of smart data sharing. I say smart because a lot of this data sharing needs rich metadata with it, so for downstream analysis, and sort of a standalone passable data sets is ideal. And number three is a common spatial biology language, cell ontologies and controlled language of how we describe the biology and the cell types is really important. So if we can get this in place as a community, I think then scientists don't have to fuss as much and concentrate on looking at biological insight.
I want to thank the panel again for the wonderful discussion today, and I want to thank all of you tuning in and watching us today. I encourage you and welcome you to come visit nanostring.com to see the power of genomics and CosMx in action, both with the live content that's available on the website, such as Minerva Stories and the interactive viewer for the CosMx data, or to download and explore the data at your own leisure using the open source tools that we have available on Bioconductor. And with that, thank you so much for your time. Please be sure to check out the concurrent session that was also filming during this time on spatial biology.